All right. Hello, everyone. We will be starting. Thank you so much for coming to this Invasive Crayfish Collaborative webinar, the first of many that we will be hosting over the next few months. So my name is Natalia Shklaruk. If some of you guys don't know me, I am with Illinois Indiana Sea Grant and am the new main facilitator of the Invasive Crayfish Collaborative, which is basically a program that brings together a variety of experts and stakeholders to address the threats of invasive crayfish. And one of the roles as the ICC facilitator is to help share information about new invasive crayfish work being done, and to also address the ways that we can all collaborate to prevent their harmful impacts on our ecosystems. So before we begin, I wanted to give a list of different links that you can follow if you're interested in learning more about the ICC. You can follow our website, the invasivecrayfish.org. If you're interested in joining the membership, you can follow these links, and hopefully that'll be added to the chat sometime so you guys can just copy and paste those. And if you're interested in registering for any future webinars, we have that on our events page. Or if you wanted to see past webinars, we also have some of those. Or if you have any other questions or comments, you can email me at shklaruk@illinois.edu. With that, one of the goals of the ICC is to make new information regarding invasive crayfish more accessible, which in turn will make people more informed, enabling them to make better, more informed decisions about invasive species management. And we all know that collaboration and information sharing often leads to more effective solutions. Different perspectives and expertise contribute to a well-rounded approach to problem solving. And in this case, the problem that we are all trying to solve is the problem of invasive crayfish. So in today's talk, we will learn about the work being done at Loyola University Chicago. Dr. Reuben Keller will discuss the recent research he and his lab have conducted on the food web impacts of invasive red swamp crayfish and rusty crayfish in the Chicago River and Lake Michigan. Dr. Reuben Keller is a professor in the School of Environmental Sustainability at Loyola University Chicago and specializes in freshwater ecology, invasive species, and bioeconomics. He leads an aquatic invasive ecology research lab that focuses on topics such as the distribution and impacts of native and invasive crayfish, barriers to spread, risk assessment, and human welfare impacts. And he spends a lot of time meeting and working with policymakers at the city, state, regional, and national levels to ensure that his research addresses important questions and that his results are communicated to those who can use them. He also holds a PhD in ecology from the University of Notre Dame and was also a wonderful master's advisor. So thanks so much for being here, Reuben. So just for everyone joining us today, we encourage you to type in any questions that you may have in the Q&A box at any point during the presentation, and then eventually when the presentation is over, we will go through them and answer them then. And also this webinar is being recorded and it will be posted to the ICC website with captions shortly after. All right, so with that, Reuben, you can go ahead and share your screen.
All right, thanks, Natalia. Is everybody seeing my screen?
Yep, it's working on my end.
Sounds good. All right, thank you very much, Natalia. As she mentioned at the very end, Natalia recently graduated with a master's degree after working with me in my lab for a long time as both an undergraduate and a master's student. And it's wonderful that she has decided to stay in the region and to keep doing good work and to keep working on crayfish. So I'm going to talk today about work that that my lab has been doing. And if I ever accidentally say what I really mean is we, me and my lab, looking at the food web impacts of the two invasive crayfishes that we find in the Chicago region. So those are the red swamp and the rusty crayfish. And this work has been funded by the Illinois Department of Natural Resources and U.S. Fish and Wildlife Service, And a lot of what I'm going to be presenting today in the food web section is work that is being led by my current master's student, Grant Lesk. Okay, so this all began back in 2015. I was still a relatively new professor here at Loyola University Chicago, and I really wanted to set up a field system. I was able to get some money out of the Illinois Department of Natural Resources, and that money was initially to spend a summer sampling for crayfish throughout the Chicago region. So when I began that, the first question was, well, what do we know about crayfish in this region? And it turned out that even as late as 2015, we knew very, very little. There were only a handful of samples from around the area. So we didn't really know what species were out there, and we didn't really know where they were. So from 2015, and this is continuing now, this map that I'm showing is from 2021. This was all of our data up till that point. But from 2015 onwards, we have been sampling crayfish in rivers, lakes, streams, ponds, and of course, that very large lake, Lake Michigan. So we've been trying to get at this question of which species are out there, and where are they, and what sorts of densities are they in. So we sample a bunch of different habitats. We use a bunch of different methods. And what we've found is that the really big story out there is rusty crayfish. We find them in almost every habitat that we go to. And when we find them, they are almost always the dominant species. So that's shown in this top left panel. These are the places where we have found rusty crayfish. And you can see the legend here tells you what types of habitats they are. But again, rusty crayfish, they're an invasive species. They're found throughout the region. They're found if we're out scuba diving in 40 feet of water. The most likely next crayfish species that we will find will be rusty crayfish. If we're in a small mucky pond somewhere, then the most likely species we'll find is rusty crayfish. They're a really remarkably adaptable species at high densities throughout much of this region. The White River and the Northern Clearwater crayfish are not shown here. They were each found at single sites, but the other species that we found are the red swamp crayfish, which we believe is a much more recent invader. It's found at fewer sites. The virile crayfish is a native. It's really widespread, but we only ever find it at low densities. And the calico crayfish is not particularly widespread at all. We only find it in this area of the north branch of the Chicago River, but where we do find it, we can find it in reasonable densities. So for the first part of my talk today in Project 1, I'm going to talk about the food web impacts of rusty and red swamp crayfish in the North Shore Channel. So what we discovered, and we found this in 2015, and we've been sampling it every year since, so we know that it is still true, is that rusty and red swamp crayfish have a fairly long area of overlap up here, several miles over which we find rusty crayfish and red swamp crayfish coexisting. And this is really interesting. These are two of the biggest, baddest invasive crayfishes that are known anywhere in the world. And one of the hallmarks of when they normally turn up and invade a system individually, when either rusty or red swamp invade a system, one of the hallmarks is that the other crayfish generally disappear. So we got really excited. Well, I got really excited when we found these overlapping populations, because I thought, wow, we must have captured them right at that point where one of these species is going to take over, right? Because that's what they do. They get into a new habitat. They're so invasive that they extirpate the other species. But instead, what we've seen over time is that these two species are coexisting, and they seem to each be doing relatively okay in the presence of the other. So I'm going to talk about work that we've been doing looking at their diets, trying to figure out how their populations are overlapping. Are they petitioning their feeding niches? And also looking at how predatory fishes might be affecting them. And in project 2, which I'll talk about later, but I'll just introduce it now. The other thing that we noticed, or one other thing that we noticed when we were doing our initial sampling, is that if we look offshore in Lake Michigan, that's where we find rusty crayfish at really high densities too in some subhabitats. So we were interested, and I'll talk about this in the second half of my presentation, in looking at distribution and food web impacts of rusty crayfish in Lake Michigan. But first of all, project 1, looking at the North Shore Channel. So just to orient everyone, downtown Chicago is right about here. The North Shore Channel, which is zoomed in on this right panel, is a canal that was dug. It was open in the 1920s. And it was created to do what Chicago was very concerned about doing at the time, which is getting rid of its sewage, essentially pushing its sewage down to the Mississippi River. So there is a large sewage treatment plant right here at Howard Street, and the North Shore Channel was constructed so that water could come from Lake Michigan, move through the canal, dilute what was coming out of the treatment plant, and then keep pushing it down into the Chicago River, and then from there out towards the Illinois and the Mississippi River. So that's why the North Shore Channel exists. It is a canal. And what we have found as we've sampled it over the years is that at the north end, we find that the crayfish populations are dominated by rusty crayfish. At the south end, they're dominated by red swamp crayfish. And in between is where we get most of the overlap, although we do have some rusties that we occasionally catch way down here, and very occasionally we catch a red swamp up in the far northern part. So we go back to the North Shore Channel. Well, we're there almost constantly during the summers, but one of the things that we do every summer is what we call our whole channel trapping. And we go along the whole channel and we lay traps so that we can get an idea of where these species are within the channel, the rusty and the red swamp. So these maps and the maps that will be on the next slide are put together in the same way. This shows for one of our sampling efforts, I believe it was in 2021, on the left-hand side, each dot is a trap that we set, and it is colored by the number of rusty crayfish on the left-hand side that we captured. So that's always on the left-hand side of the channel. On the right-hand side is exactly the same, but colored by the number of red swamp. So where it's a white fill, we found 0. Where it's a yellow fill, we found 1. Orange is 2, red is 3. So you can see really clearly here that at this north end, up closer to Lake Michigan, dominated by rusty crayfish, not very many red swamp, or 0 during this year. As we come down, we have this zone of overlap. And then as we move further south, and this is getting way down south, we get to this area where it's predominantly red swamp crayfish. So for the analyses that I'm going to be talking about, we designated these kind of creatively as Zone A, which is where rusty crayfish are dominant, Zone B, where there is pretty even overlap between these species, and Zone C, which is where the red swamp crayfish are dominant. And the question that we wanted to ask was, well, what's going on with feeding by these crayfish in these different zones? And we thought, well, I should say I thought, I'm not going to ascribe this to my lab because it, as you'll see, it turned out to not be completely correct. I thought that rusty crayfish would have a higher trophic position when they were found without red swamp crayfish around. Red swamp crayfish are larger crayfish, they're more aggressive, and in lab experiments, we've seen that they generally, when we do that classic experiment of 2 crayfish, 1 piece of food, the red swamp crayfish generally gets the food. So my hypothesis was that rusty crayfish would have a higher trophic position they'd be eating better food, essentially, where there were very few red swamp crayfish around, and they would have a lower trophic position where they overlapped with red swamp crayfish. So that was the sort of the hypothesis behind that first study, where we've used stable isotopes to look at trophic positions of crayfishes in those 3 zones. We were also really interested in, well, we know that fish eat a lot of crayfish, and we're interested in whether that might be somehow driving this pattern, these patterns of distribution that we're seeing. So we've done a lot of work looking at fish diets via lavage, and I'll talk about that in a moment. So for the stable isotope analysis, we collected a lot of crayfish from zone A, a lot of crayfish from zone B, and a lot of crayfish from zone C. This photo shows an undergraduate in my lab, Michaela. She is dissecting muscle tissue out of a crayfish. We take the muscle tissue out, we dry it. That's shown in this middle photo. And then we grind it up and we put a very small amount of it. We very carefully weigh a very small amount of it into a little capsule. And then we fold that capsule up. We send it all off to a lab at University of Georgia, and they look at stable isotopes. And from those stable isotopes, we can tell whereabouts on the, or we can tell what trophic position each individual crayfish was living at. So essentially, we would expect that if crayfish are getting the good foods, this means they're eating higher up the food chain, they would have a higher trophic position. If they're eating further down the food chain, they would have a lower trophic position. So I'm going to cut straight to the results here. And this is going to take some explaining because there's quite a bit going on. So on the x-axis, we have our delta C value, and on the y-axis, the delta N. The delta N from stable isotopes is by far the most important. That tells us the most about the trophic position at which the organisms are existing. Each triangle is a red swamp crayfish, Procambarus clarkii. Each circle is a rusty crayfish, Faxonius rusticus. And then we have our 3 communities divided by color. So a purple circle, this is a rusty crayfish from community A, which is the furthest north. A blue circle is a rusty from community B. A blue triangle is a red swamp from community B, and so on. So what this tells us is that there is no difference, no noticeable difference, because these all overlap so strongly between communities A and B, and that it doesn't matter whether you're a red swamp or a rusty crayfish. The trophic levels over which those 2 species are existing in community B is basically identical. And in community A, where I thought that those crayfish, the rusty crayfish, would have had a higher trophic level, they don't. The only place that we see a difference is when we look at red swamp crayfish down in zone C. And this is where they are living by themselves and there are no rusty crayfish. Down there, the trophic position is quite a bit higher. So this looks like an exciting result, but I'm sorry to say that we don't think that it is. We think that what's going on down here is that there is a lot of nitrogen enrichment from the sewage treatment outfall. So we think that the sewage treatment plant is, well, we know that it is putting a lot of nitrogen into the system, and that is driving these delta N values higher. So we actually, it's very difficult to, well, it's impossible really to compare these values up here to these values down here, because these were collected from water that has such a different level of nitrogen. But the short story of this is, I think, that a crayfish is a crayfish. In terms of trophic level, it doesn't matter whether it's a rusty or a red swamp. And it doesn't matter whether they're living by themselves or living in a community with the other species. Their trophic level is very similar. And that's emphasized here. This is really just showing the same data, but in a slightly different way. This is looking at trophic position on the y-axis This first one is rusty crayfish in zone A. Then we have red swamp crayfish and rusty crayfish in zone B. And you'll see that their trophic positions are very, very similar there. And then we have red swamp crayfish in zone C, where the trophic position is higher. But again, we believe that that's more of a result of the confounding variable of the sewage treatment plant outfall. So, okay, so I'll finish this section with that. We really don't see a difference. It doesn't seem to matter in terms of diet. It doesn't seem to matter whether you're a red swamp crayfish or a rusty crayfish. It appears that you're eating very, very similar foods out there. And it also doesn't seem to matter whether you're living sympatrically with the other species or whether you're living by yourself. These crayfish are eating similar types of food. The next part of this project has been looking at the predators of crayfish in the North Shore Channel. And this has been a lot of fun. We go out with the Fish and Wildlife Service or the Illinois Department of Natural Resources, occasionally with the Army Corps of Engineers with their electroshocking crews. And they go out every spring and fall, and they're doing the sampling to look for Asian carps. So they're catching a lot of fish via electroshocking. They give us their fish, and we lavage them. So we get these living fish and some pretty fantastic largemouth bass, as you can see here. And we lavage these fish, and I'll explain what lavage is on the next slide. On the left-hand side here, we have Carter Cranberg. He was a lab technician with me when we started this work. He's since gone off to work at the EPA. In the middle, we have my son, Freddie. Two of his great loves in the world are getting a day off school and fish. So he was particularly happy when I told him that he could come out with us for a day. And on the right-hand side, we have me and Carter's lower body. This shows you the basic setup. Carter would hold the fish, and I would lavage them. We would collect what came out of them, and then we'd move on to the next fish. So this is what the canal looks like. This was with one of the DNR fishing boats. And this is what lavage looks like. It's basically as straightforward a procedure as it sounds. We take a tube, we stick it far into the throat, all the way into the stomach of these fish. We open up the tube so that water flows into the stomach, and that basically flushes the stomach contents out, which we capture in a strainer down here, and then we can have a look. And what we're most interested in with this work is seeing if crayfish are being eaten. If they are being eaten, do these fish seem to have any preference, or are we finding more red swamp or more rusty crayfish in the fish? And what might that tell us about, what's going on with the balance of the rusty and the red swamp crayfish that we're observing? So this is what the stomach samples look like. We can find, bits of crayfish like this. This is a piece of carapace that was all that was left. We can find bits of walking legs and broken up bits of carapace, or we can find a nice chelae like this and all of the walking legs. But generally, we find pieces of crayfish in various states of decomposition. So to date, we have sampled, and sampled means that we have made an attempt to lavage 456 fish. From those, we have collected 244 samples. So that means that roughly half the fish, we lavaged them and nothing came out of their stomachs, meaning that they just didn't have anything. They hadn't eaten anything recently enough. But for roughly half of them, 244, we were able to collect samples. From those samples, we have been able to identify that 70 contained crayfish. And this is the really remarkable part. 68 of those crayfish have been red swamp crayfish, and only 2 have been rusty. So 68 red swamp, only 2 rusty. So almost all of the crayfish that we have found in the stomachs of these fish have been red swamp crayfish. And we it turns out that fish, this might not be too surprising, but fish are much better at finding red swamp crayfish than we are. Again, these figures have quite a bit going on. The triangles are our sampling. So this is our whole channel sampling. So where you see a blue triangle, that means that we've found a rusty crayfish there. Where you see a green triangle, that's where we found red swamp crayfish. The circles represent our results from lavage. So a yellow circle indicates that we found a fish with one red swamp crayfish in it. A red circle means that we found a fish with two red swamp crayfish in it. So if we concentrate first just on this area way up in the north of the channel, you can see that, you know, we as scientists with our trapping, we can't find red swamp crayfish. And yet, if we find crayfish inside a fish, it is always red swamp crayfish. So this is one of the two rusty crayfish that we found, was down here at Green Bay, on Green Bay Road. But still, where we were getting crayfish out of fish, even though the crayfish community through here is dominated by rusty crayfish, or at least, you know, a shared dominance between rusty and red swamp. Almost all of the fish, 68 out of, almost all of the crayfish, 68 out of 70 that we recovered from fish stomachs were red swamp crayfish. And this is true as we go further and further down. This was the other red swamp that we found down where the north branch of the Chicago River meets the North Shore Channel. But the story here is very much that fish are preferentially, or perhaps preferential is the word, is not the word, but where we find crayfish inside fish, it is almost always red swamp crayfish. And that is true regardless of the relative densities of red swamp and rusty crayfish. So for some reason, red swamp crayfish are turning up in fish stomachs. They're being consumed by fish at a much, much higher rate than rusty crayfish. So this again sort of gets us towards thinking about what's going on in the food webs and what the relative impacts of these two species might be. And we have some other work, which I'm not going to talk about today, but we have some other work, and this kind of gets at it, which indicates that the reason that red swamp and rusty crayfish are able to occur sympatrically, that you can have their populations overlapping. A potential reason for that is that, well, the red swamp crayfish are competitively dominant, right? They will outcompete rusty crayfish for resources, perhaps space, certainly for food. But to be able to do that, they've got to be really aggressive, and they've got to be out there, and they've got to be always ready to fight. That might be the reason that the fish are eating so many more red swamp crayfish, because the red swamp crayfish are more active, they're more aggressive. So that's our working hypothesis at the moment, is that these two species are overlapping because even though red swamp crayfish is competitively dominant, that comes at a price. So red swamp crayfish are winning battles with rusty crayfish, but they're losing battles with largemouth bass. So I didn't talk about this yet, but it is large. I just mentioned it. It is largely largemouth bass that are eating the crayfish. There is a really like a pretty fantastic population, particularly for an urban area, of largemouth bass through the North Shore Channel. And that's because there's very little access to the channel, so there's very little fishing. We were interested in whether the size of the largemouth bass made a difference to whether or not they had crayfish in their stomachs. So on the x-axis here, we have the fish length in centimeters. So we divided them up into 7 bin sizes here, from a pretty small largemouth bass to a really large one. And what we found is that it doesn't really seem to matter how large the largemouth bass is. They consistently about 15% of the fish in each size class that we bring back, or that we lavage, consistently about 15%, although a little less for the small ones. But that's the percentage that have crayfish in their stomachs. At these very large sizes, we just didn't catch enough up there. We had 4 in each of those two largest size classes. So we just didn't catch enough to say anything particularly conclusive up there. But it doesn't seem to make a big difference how large the largemouth bass are, except for the very small ones tend to not have, or are less likely to have crayfish in their stomachs. So in conclusion, rusty and red swamp crayfish have overlapping populations, and we don't see any sign of trophic niche petitioning. They seem to be both feeding on much the same stuff, right? We're not seeing that one of them is existing at a higher trophic level. And that's what we learned from the stable isotope analysis. We found that largemouth bass are the main predator of crayfish in the North Shore Channel, and that they are taking almost entirely red swamp crayfish. And I spoke about our working hypothesis for that, which is that red swamp crayfish are persisting because they are more aggressive than rusty. So they're able to outcompete rusty for a lot of resources, but that the aggression required for that makes them more susceptible to being taken by largemouth bass, and that is a depressing force on their population size. Okay, moving on to Project 2. So this began when we started doing work out in Lake Michigan, scuba diving and catching crayfish. And what we found out there was, as I said, rusty crayfish. And in good habitat, in good rocky habitat where crayfish have a lot of places to hide, we could find a lot of rusty crayfish. And this got us interested then in thinking about whether rusty crayfish are existing at a different trophic position to native crayfish. So as I said, we began all of this work by looking at what was known about crayfish distributions around the Chicago region. And prior to 2008, there were only 77 records of crayfish from Lake Michigan, from the Illinois portion of Lake Michigan. And almost all of those were from beaches. So that means that somebody had been walking along a beach, they'd found a dead crayfish washed up, they'd been able to identify it, and they'd reported that. But there were only a very small handful of records of crayfish that were taken from offshore. We knew that historically the native species have been the northern clearwater, Faxonius propinquus, and the virile crayfish, Faxonius virilis. And we know that the first record of rusty crayfish in Lake Michigan in this area was in 1984. And from those 77 records, there was some evidence that rusty crayfish had been spreading between then and 2008, but not really clear evidence in the form of, repeated sampling, lots of sampling from different areas of Lake Michigan. So we came to this work in 2015 and we started scuba diving and we found rusty crayfish in abundance. And that got us thinking, well, what was it like before rusty crayfish? And as I said, there were very few sort of point data available. But I was lucky and I came across an old master's thesis. This was written by John Quinn and his advisor, John Jansen, who were actually at Loyola University back then. John Jansen is still working on similar issues. He's up at University of Wisconsin at Milwaukee now. So from 1979 till 1983, Quinn and Jansen went out and looked for crayfish at 38 sites. What we know about where they went is that they went from Montrose, so this is down about 8 miles north of downtown Chicago or so, to Waukegan. Waukegan is just about at the border with Wisconsin, so this is a long area that they sampled across. So we know that they sampled at 38 sites. We know that from the thesis, we know that the sampling was most intensive between Hollywood, which is a street, and Wilmette, which is a town up here. And this is, for reference, this is the North Shore Channel here. So we're in a very similar area. So we know that the sampling was more intensive in this dark green area, less intensive in the light green area, but we don't know what that means. We don't know how many of the samples were between Hollywood and Wilmette, how many were south and how many were north. And this was in the era before GPS. So we don't know exactly where these samples were taken from. We do know that they used 50-meter transects. So they would find what they thought was a likely place for finding crayfish. They would drop a 50-meter long rope. They'd stretch a 50-meter long rope along the bottom. They would jump in at one end, go to the bottom, scuba dive along. And as they were going, they would collect as many crayfish as they could from 1 meter on either side of the transect. So we know how they sampled. We don't know exactly where, but we've got a pretty good idea. And we know that the only crayfish that they found were the virile and the northern Clearwater. So over this 4-year period, sampling at 38 sites, they only found the natives. So this was actually a pretty remarkable piece of work for us to find, because it allowed my lab to then say, well, we can go back to this area, and at least at one point in time, we've got a pretty good idea of what was there. So we can now go back and see how populations of crayfish in this area have changed. It's worth mentioning also that Quinn and Jansen were not particularly interested in crayfish as crayfish. They're interested in crayfish as food for yellow perch. So the whole piece of work there was done to look at what food was available for yellow perch, as opposed to having a deep and abiding interest in crayfish for the sake of crayfish. So this has been a lot of fun. We go back, we go out each year and we scuba dive. And we do our best to go to the sites, or the sort of sites that Quinn and Jansen were going to. We have this pretty cool experience of having the Chicago Lake, the Chicago skyline in the background, and we get to go in and dive. And when we get to the bottom, if we've chosen our site well, we use sonar to find a spot with lots of rocks, and we see scenes like this. So this is our transect rope here. And you can see that there are lots of rocks on the bottom. This is the remains of the glaciers from, I guess, about 10,000 years ago, still sitting down there. And if we find a rock like this, where there's clearly some space underneath, then we can turn over the rock and see if we can catch a crayfish that's hiding under there. You'll also notice that just about everything is covered in dracenids down there. And there's lots of cladophora around. And if we see a fish, it is almost always a round goby. So this is very much an invasive species dominated habitat now. And yes, when we get lucky, we find a crayfish. This is Thomas, who came out with me and dived several times over a summer. And he's holding up a rusty crayfish that he just caught on the bottom down there. Okay, so this left-hand figure is the one that we've seen. This describes where Quinn and Jansen did their sampling. The right-hand figure, I've zoomed in here just from Montrose, Wilmette, and then a little further north. And I'll explain the reason for that in a moment. So when we started at Montrose, and if we head north, then we find only rusty crayfish, and that's the red dots. A red dot is where the only species that we found along the transect was rusty crayfish, FR for Faxonius rusticus. So we sampled along there for about 1.5 years, and it was just rusty. And then as we headed further north, we had what to me was an incredibly exciting experience of starting to find some native species. So these purple sites, 5, 7, and 9 here, are where we found northern Clearwater crayfish, Faxonius propinquus, and rusty crayfish at the same sites. And if you can imagine the scene, you're down there scuba diving, you're very much in your own little world because it's very difficult to communicate with anyone else. The visibility is not particularly good. I can vaguely see my dive buddy who might be 10 feet away. I can sort of see the cloud that is, or the shadowy shape that is them. And I suddenly realized that I picked up a native crayfish. And then I picked up more and more, and it was just incredibly exciting. I was, yelling and screaming into my regulator. It was it was very cool. So we find this overlap up here north of Wilmette between rusty and native crayfish, northern Clearwater. And then if we go further north, we only find native crayfish. And that is true all the way up to Waukegan. So our sampling north of up towards Waukegan has been a lot more sparse because there is a lot less habitat up there. But everywhere that we have looked up in this most northern region is only the native species. So what we found, to summarize, is rusty crayfish dominating up as far as Wilmette Harbor, a zone a couple of miles of shoreline where there is overlap, and then only natives beyond that. So this is certainly a very, very large change from what Quinn and Jansen found when they only found the native crayfish. So we wanted to look at, again, I'm talking about food webs, we wanted to look at trophic positions of rusty crayfish when they are existing by themselves at a transect, where they're existing overlapping with native species. And then we wanted to look at trophic positions of native species as well. So again, back to our stable isotopes, and this time we've defined community 1 is where we only find natives. Community 2 is where we find natives coexisting with rusty crayfish, and community 3 is where we find rusty crayfish only. Same procedure for stable isotopes. And once again, we find a mess of data. So the circles are northern Clearwater, Faxonius propinquus. Triangles are rusty crayfish. The squares are the virile crayfish. So circles and squares are the natives. If they're colored red, then it's an area where we have natives only. So we get a large range of trophic positions. And again, we're looking mostly at the y-axis here to see where these things are falling trophically or how they're differentiating trophically. The green is where we have rusties and natives. So that's where I really thought, again, that the rusties might be pushing the native crayfish down on the trophic scale. If that were the case, then all of these green triangles would be higher than all of the green circles, but we certainly don't see that. And blue is where we have rusty only, and that's sort of spread throughout this area. So we don't see any evidence here at all that there is any sort of trophic petitioning going on, which is a fancy way of saying we don't see any evidence at all that these different species in different communities are eating different foods. And just to emphasize that, we can put those ellipses on again, which show, in this case, they show a 40% confidence interval for where these crayfish are in the in this space, in the delta C, delta N space. And every one of these ellipses overlaps with every other one. So it's remarkable, I think, that that so little is going on with this. And in terms of trophic position, again, these are very, very similar. Propinquus, the northern Clearwater, and the rusty crayfish actually have exactly the same mean trophic position. The virile crayfish is slightly less, but this is just a tiny, insignificant difference. So again, out in Lake Michigan, a crayfish is a crayfish. They all appear to be persisting on roughly the same diet. So we don't see an effect, really, of this transition from the native crayfish to rusty crayfish. We took one further step and we said, well, maybe the rusty crayfish are existing at higher densities. So maybe the rusty crayfish are having impacts in that way. So we compared the total number of crayfish per transect. So each transect was 50 meters long. We captured all the crayfish that we could within 1 meter of the transect line. So each transect is 100 meters squared. So we compared the total number of crayfish per transect at sites with and without rusty crayfish. Very straightforward t-test, and we found that there was no significant difference. In fact, not even close to a significant difference. Now, of course, this is a somewhat confounded test. It's confounded by habitat. All the rusty crayfish sites are down in the south, all the native sites are up in the north. So this is a somewhat confounded test. So it's not particularly strong, perhaps, but still, we don't see any impact there of rusty crayfish on the total number of crayfish. So in summary, what we have seen is that rusty crayfish have replaced native species over a large area. And because we were able to find and then use these data from Quinn and Jansen, this is really one of the first places that we've been able to sort of conclusively say that rusty crayfish have replaced the native species, and we can at least put a time bound around when that happened. There's no significant difference in the number of crayfish, regardless of the community, and the trophic levels of all species are similar, regardless of community. So it appears that the rusty crayfish are eating pretty much the same food as the native crayfish that they've replaced. And that's the end of my presentation. Thank you very much. And I am happy to, well, I believe that Natalia is going to moderate. She will tell me how I would like to take questions.
Yes, so we have a few already. I can read them out to you. So there's one question that asks, did you ever find rusty and propinquous hybrids?
We know, yeah, that's a really good question. We know that those have existed. They've been well described by Bill Perry in inland lakes. Bill has done some work in Lake Michigan. We looked for them morphologically, which is crude compared to looking for them genetically. So for some background, we know that rusties and northern clearwater do hybridize in some situations. The short answer is no, we did not find any hybrids. But I'm not going to, but we were not doing the genetics on them. So it's possible that those hybrids do exist. But every crayfish that we've found out there, we've felt pretty confident saying that's a rusty or that's a propinquous. We have not seen crayfish that have, morphological signs that they're a hybrid. So I can't guarantee that they're not out there, but we haven't seen those signs that we would have been looking for.
Okay. We have another question here. Have you ever tried to estimate the pairwise probability of groups overlapping? And I think this is referring back to the stable isotope graphs that you were showing.
Pairwise probability of groups overlapping. No. And I'm going to plead a certain level of ignorance here. Those results came from my graduate student grant. We recently got those results back, so I lent on him pretty hard to put together those graphs for this presentation. But no, we have not done that.
Okay, great. Another question in the chat. What are the predators of rusty/red swamp crayfish outside of the largemouth bass? Do seagulls eat them?
I am sure that birds take them. I suspect that seagulls are not a major predator. I'm sure that we see a lot of herons along the North Shore Channel. So I suspect that they take crayfish. There are muskrats, which may take crayfish as well. I wouldn't be super surprised if, the occasional raccoon gets in there. There are places where, particularly, at night, it's not too difficult to see the crayfish running around or, the crayfish swimming around under the water. The other fish that we find that have crayfish in them, we don't get many of these, but catfish and bullhead quite often have crayfish in them when we find them. Crappie, again, we don't get many crappie, but they do quite often have crayfish in them. One of the, there are a lot of panfish, or particularly bluegill, through the system. And we know from some other systems that they can eat a lot of small crayfish. We were not able to effectively lavage those bluegill. And those that we felt like we could effectively lavage, we did not find crayfish in them. So I'm hesitant to say that they are not eating crayfish because I don't think we did a good job of lavaging them. But we did do a good job of lavaging some of them, and we didn't find crayfish. But yeah, those are what I think are the other predators of crayfish out there.
Great, thank you. Also, what kind of traps and bait did you use? Was asked by one person also.
For the crayfish, we used G minnow traps, the standard minnow trap. We enlarged the openings a little bit, which is just sort of fairly standard for crayfish. And we use hot dogs as bait. So another project which is ongoing is trying to reduce the population of red swamp crayfish in the North Shore Channel. So we have right now a couple of hundred traps out there where we are trying to catch as many red swamp crayfish as we can. And we've been doing that. I think we're just finishing up our 6th season of work doing that. And along the way, we have done a lot of work to try and become better at trapping crayfish. And one of the things that we've discovered, and we've done this experimentally, and we have good results to support this, is that at least in this system, and at least for red swamp crayfish, we catch about twice as many crayfish when we use hot dogs as bait as for any other bait that we've tried. We've tried dried dog food, dead fish, dead crayfish, beef liver, and one other. I remember that we had 6 different baits that we tried. But hot dogs captured roughly twice as many red swamp crayfish as anything else that we tried.
Great, thank you. The non-natives don't seem to shove natives or each other into lower trophic levels. Is one potential reason for the high level of invasives relative to natives perhaps habitat limitations, particularly in the the Chicago area waterway system?
Habitat limitations, I'm not sure I understand that part of the question. If that person wants to unmute and clarify, I'd be happy to.
Yeah, sorry, I'll go ahead. I've got a lot of thoughts rattling around. So just noticing that in your sampling, the further inland in the river system you seem to go, it seems like the invasives become quickly much more dominant, red swamp in particular. So I'm wondering if do these have different habitat niches in terms of species, or is most of their overlap just going to be competing for their food web position, I guess is the simpler way to ask that.
Yeah, and the really simple answer is I don't know. We have done, this is a canal. It is really homogeneous in terms of, at least to me. I'm obviously not a crayfish, but to me it is homogeneous substrate. It is certainly homogeneous depth throughout the system, and we've done some work. So that was actually, we know that red swamp crayfish, at least in a lot of habitats, they like to burrow. So we did some work trying to compare the extent to which it was easy to dig a hole at different points along the canal, and we could not find a significant difference that was associated with we could not find a significant difference, period. We certainly couldn't find anything that was associated with that transition that we see from rusty crayfish being dominant up at the top of the canal and red swamp crayfish being dominant further down. The one big difference, and I actually think that this is actually, it's a habitat difference, so it's exactly the type of thing that you're suggesting, is that where we find red swamp crayfish at the highest densities begins about a mile downstream of the sewage treatment plant outfall. My thinking about that is that the sewage treatment plant outfall keeps the water down there, downstream of it, warmer during winter, whereas upstream it's cooler. So I think that it might be a temperature effect. So rusty crayfish, their native range is around in the Ohio River Valley, but they're certainly capable of surviving, well north of Chicago. Red swamp crayfish's native range is, down in Louisiana and northeast Mexico. So they might be really stretching their thermal tolerance to survive where they are. And that might be what's, it's obviously not killing all of them, because we do find them, upstream of the sewage treatment plant, but it might be what's allowing them to survive in really high densities down there. So actually this winter, I have a student who's about to do it. We're going to be putting eye button temperature loggers out all the way along the channel to look at how strong that effect of the sewage treatment outfall is in terms of raising the water temperature downstream.
Great, thank you so much. Next question. We have a few more, so I'll try to get to as many as I can. But this next question, do the bass tend to stay in their area of the river, or do they swim up and down the channel? If so, could they be eating the red swamp crayfish from down south, then swim up the channel?
We don't know, and yes. There is the Shed Aquarium is doing some work. They have radio tagged some fish, and we're going to be really interested to see the results that come back from that. So they've radio tagged some bass. They're not looking at it up in our area. They're looking at it further downstream in the Chicago River, but we're going to be interested to see how much movement there is in the bass. But bass, it's possible that there's a whole lot of movement going on, and that's so, and that's why we're finding bass with red swamp crayfish in them in an area where we can only trap rusty crayfish. And that's a possibility.
Great. Are there enough predators to manage these invasives?
More predators would be better. And part of the way that we always tell a story when we're applying for money. A part of the story that I told, which I genuinely believed at the time, was that if we could find out what type of fish are consuming crayfish in this system, and also what size, then we could try to enhance those populations. So we've certainly found that red swamp that the largemouth bass are by far the biggest consumers of red swamp crayfish. And there are so many, it's a pretty amazing fishery for largemouth bass. I don't think you could squeeze more largemouth bass in there. So I think that if there were going to be efforts to enhance predators, then it should come from the other species that we find that I think there might be some more niche space for, would be bullheads and catfish. And the Friends of the Chicago River have programs to release catfish, and so we're hopeful that that might help. Because we get our fish from electroshocking, and electroshocking is not super effective for catfish and bullheads, it's possible that there's a much larger population of them out there than we know, and they might already be eating a whole lot of crayfish.
Great, thank you. Let's see. What was the spacing between the minnow traps in the canal?
It's so throughout the system, it's always either 50 meters or 100 meters. We can't do 50 meters for the whole canal just because it would take too long. So for those areas where we know that we have, well, basically from the northern end and working down for about a third of the way, we space them about 50 meters apart. South of that, there are fewer crayfish. So we space them 100 meters apart for quite a long stretch. And then we go back to 50 meters apart further south. Yeah, so we've sort of settled on that as being an arrangement that makes it possible for us to get the work done to cover the whole of the canal. And that gives us good resolution in those areas that are most interesting in terms of monitoring the spread and density of these two species.
Great. And last question. Based on your results, what recommendations do you have for conservation organizations concerned with the invasive crayfish?
Yeah, I mean, I think, speaking very broadly, we've got to stop these species from being moved around. We've done a huge amount of work, as I said, trying to reduce the population of red swamp crayfish. And we can do it, but it takes, we're working at it every summer. And at least with the technologies currently available, there's no way to eradicate red swamp crayfish from this canal. So the biggest bang for the buck is preventing the spread of these species, and particularly red swamp crayfish, because it is a relatively new invader. Rusty crayfish are still spreading, but to a much larger extent than red swamp crayfish. Rusty crayfish are all throughout the system, or throughout the systems in this area. So it's, we're never really going to get rid of them. So the biggest bang for the buck is preventing the spread of red swamp crayfish. Our work shows that in the North Shore Channel, we can reduce the population of red swamp crayfish. And we think that that is really useful because it does reduce the chance that they're going to spread. It reduces the chance that they will spread, sort of naturally. It also reduces the chance that people will be able to catch them and perhaps use them as bait and move them around that way. We have colleagues in Michigan who are working on red swamp crayfish invasions in ponds and small lakes. And they have a lot more options available to them in terms of control, because they tend to be working in retention ponds. So it's okay to poison a retention pond and try and kill everything, including the crayfish, right? It's not okay to do that in the North Shore Channel. But we're talking to them a lot about our results with the predatory fish, and they're doing their own really good work looking at introducing fish into some of the ponds to act as a help. So there's several sort of related things for conservation organizations, but I think that the biggest bang for the buck by far is preventing the spread of these species.
Yep, prevention is best. Okay, so that is all the time that we have today. If you guys had any other questions, or I didn't cover a question that you asked in the chat or the Q&A box, you can email me them, and I'll make sure to forward it to Reuben, who will then contact you and answer you then. So again, thank you so much, Reuben, for giving us your time and presenting the fascinating research that you and your lab are working on. And thank you to everyone who attended this meeting. And again, to remind everyone, this recording will be posted on the ICC website with captions. And our next ICC webinar, it'll be held later in October, and I'll send more info with that coming soon. So thank you again so much for being here, and I hope everyone has a great rest of your day.